So they went on to break it down further by gender, sport, and ethnicity, but the main thing that I want everyone to take away from this is that at least 22 million Canadians are involved in sport. And with that, it's understandable why it's garnering a lot more attention, and while there's so many benefits, there are risks associated with it. For example, in contact sport, the contact can be head-to-head with another player, to the body during a tackle, or with the ball or a puck, all of which have the potential to cause injury. In our lab, one of the major focuses is brain health. And to maintain good health, we ideally want to avoid injury, especially considering that the cost for brain injury cost the Canadian healthcare system about $3 billion annually. So there's huge economical value behind this research. So traumatic brain injuries, or TBIs, are one of the most common neurological conditions that affect Canadians, about 365,000 people every single year. And they're broken down into 3 categories: mild, moderate, or severe. And they're classified clinically based on symptoms such as motor issues, confusion or memory loss, headaches or mood changes, and loss of consciousness or awareness. The difficulty with this area is that the symptoms vary. For example, headaches could easily be a symptom of any level of brain injury. And that brings me to the idea behind our project, which was to look at the effects of contact sport on brain health. And as with anything, when we want to do research, we want to make sure we have a solid background before we run studies in hopes of advancing the field. So what are the current practices for assessing brain health? We can do CT scans, MRIs, self-reporting, or for concussions, we've got the Sport Concussion Assessment Tool, or SCAT-5. So with brain injury and talks of how soon kids should be introduced to contact sport, I'm sure you all agree that the time for reliable testing is now. The problem with a lot of these is that they're either limited to an emergency room, which goes back to the economical aspect, or they're based on very subjective signs and symptoms that vary from person to person. So it's fair to say that our current options right now are not only time-consuming and expensive, but they're not as useful when quick game-time decisions are needed. So where do we go from here? So I'm going to introduce some of the methods used in our study that will hopefully become a mainstay in the future, and that involves using blood biomarkers of brain injury. So there are a lot of biomarkers that can help in assessing brain health, but there are 2 I'll be focusing on today. The first is glial fibrillary acidic protein, or GFAP. This is an intermediate filament found on astroglial cells and is released during cell death or injury, right over here. And the second is neurofilament light, which is highly expressed in large myelinated axons. So when there's any breakdown in the blood-brain barrier right around here, these markers can easily cross through and become more present in our bloodstream, and they increase in concentration proportional to the severity of damage. So they show great promise as possible indicators that brain injury may have occurred. But the issue with this is that it is still an invasive method, and our lab is looking for more non-invasive ways to assess brain health. So what other measures can be used? Well, we can't talk about the brain without talking about a certain relationship, and that is the pressure-flow relationship. So when there are any changes in blood pressure, we want to make sure that we're maintaining adequate flow, because this is critical to proper functioning. This is especially important in the brain because although in the body it only accounts for about 2% of our mass, it has a really high metabolic rate and accounts for about 20% of our cardiac output. So the idea behind what we call cerebral autoregulation is the idea of when we have a certain change in blood pressure, we will automatically change our flow, whether it's an increase or a decrease, to minimize the effect of that change in blood pressure. So this is what we call cerebral autoregulation. And there's not a lot of data currently on contact sport in terms of cerebral autoregulation. It is fairly limited, but there is one done by Bailey and colleagues over 10 years ago. So what they did was they looked at professional boxers and they compared them to healthy age-matched controls. So what they found is that when there's an acute drop in blood pressure, it happens pretty similar between groups, but the key difference is here. And you'll notice that compared to the controls, the ability of the boxers to respond to this drop in blood pressure and increase their flow to make up for that seems to be impaired. And to really drive home the point that cerebral autoregulation can be impaired by contact sport, they calculated what is called the index of cerebral vascular conductance, which is just a fancy term for asking how well do our blood vessels constrict or dilate to maintain changes in blood pressure. So what they concluded was that this dampened response was likely due to multiple blows to the head during sparring and training sessions. So with this, an increased slope basically tells us that we've got better conductance. Better conductance means more efficient cerebral autoregulation. So with this background and the blood biomarkers, that brings me to the purpose of our study, which was to assess cerebrovascular health in junior football athletes throughout a season, using the markers as an index of injury and to evaluate cerebral autoregulation pre and post-season. We hypothesized that the markers would be elevated post-game compared to pre-game, and cerebral autoregulation would be impaired post-season compared to pre-season. So in addition to these two tests, we also ran a lower body negative pressure test, a cold pressor test, and neurovascular coupling. So in the pre and post-season, we were able to do a number of ultrasound and respiratory measures along with blood sampling. During the season, blood samples were collected about 24 hours before and 12 hours post-game in the athletes and at one time point in controls. And this right here is really the novelty of this data, is that it longitudinally followed multiple players across the season, and they're instructed to just go through as normal. The players, the coaches, their medical staff were all blinded to the results. So the cerebral autoregulation was measured using what is called a 5-minute repeat squat stand protocol, as shown in the figure on the right. So the benefit of this is that it allows us to track changes in blood pressure in the hyper and hypotensive regions. And measures were also done in two arteries, the middle cerebral artery, or MCA, and the posterior cerebral artery, or PCA. And this allows us to track blood flow in the anterior and posterior portions of the brain. So what did we find? It's really important to note that there were no diagnosed concussions during the season, but across all 12 games and 14 players, a total of 97 data points were collected. So we found a significant increase in GFAP concentration post-game compared to pre-game, which equaled to about 17.7%, but there was no significant difference between the athletes and healthy controls. While neurofilament light trended toward a decrease, it didn't quite reach threshold post-game compared to pre-game, and again, there was no significant difference between the athletes and healthy controls. So this is just a portion of the cerebral autoregulation data. Today, I'll just be focusing on changes when there are increases in blood pressure. So just to orient everybody, our y-axis looks at the change in end-stage blood velocity over the change in blood pressure. So ideally, if we've got efficient cerebral autoregulation, we would have a lower value on our bars, which would indicate better cerebral autoregulation. So we found post-season compared to pre-season, there wasn't a significant difference between the athletes, but we had a significant difference between the athletes and controls, with controls exhibiting a lower change, meaning better cerebral autoregulation. In the PCA, it was the same. We also found no significant difference pre versus post-season among the athletes, but again, significantly different between the athletes and healthy controls. So with all these findings, where do we go from here? So a sign of things to come. This is an editorial that was published last year by neurosurgeons and neuro researchers in the States. And what they discussed was that brain damage to NFL players is still a big concern, and they did mention that although concussion risk has decreased since the 2015 season, it is still a major problem. So they are calling on the NFL to implement the blood biomarker testing into their head injury protocol, especially now that the FDA has approved what's called the Abbott iStat Alinity. It is a device that allows you to track the concentration of a lot of the blood biomarkers on site, and you are able to get the results within 15 to 20 minutes. So now that that device is approved, they really want the NFL to implement this, and if it is taken under consideration and it becomes a mainstay even at the professional level, it's a really good sign of things to come. So in summary, we know that across all players and games, there are acute increases in GFAP concentration, but no changes in neurofilament light pre versus post-game in contact football. Cerebral autoregulation remains unchanged among athletes, but is impaired compared to healthy controls. But the implications of this are yet to still be fully understood. So I do want to thank the collaborators at UBC and UGuelph for their help with this project, and thank you everyone